Eukaryotic DNA is found in the nucleus, and yet proteins are produced in the cytoplasm. DNA is protected inside that nucleus, and it's not allowed to leave in any way, shape, or form, so there has to be some way for the genetic information held in the DNA to get to the cytoplasm. So what we do is we first turn DNA into messenger RNA through a process called transcription. That is then allowed to leave through the nuclear pore after a few modification steps. Once that mRNA is to the ribosome, it will be translated into an amino acid sequence, a polypeptide, aka protein. So how does that DNA actually code for proteins? Well, DNA is turned into RNA, like we said, and then RNA is turned into proteins, but the actual steps are a little strange. How can you code for the 20 amino acids that are commonly occurring in living organisms with only 4 nucleotide bases? In this case, you please remember that this is an mRNA sequence, therefore there will be no thymine. It will be replaced with uracil. The actual functional segments of DNA that code for information are what we call genes, and that's probably the simplest, most straightforward definition of the gene you're going to get. So the DNA is transcribed into mRNA through the process that we saw in the last video. So what we're looking at here is how you make that final step between mRNA and a protein. What we see is that each 3 base nucleotide sequence on the mRNA is called a codon, and it codes for a particular amino acid that will be used to build the protein molecule. So if I have 4 base types with 3 positions, that means I have 64 different possible combinations. These combinations specify the 20 different amino acids used by living organisms. There are a couple of exceptions, but what we see is this overall genetic code is universal across all domains of life. Not only is the genetic code universal, it is also redundant and unambiguous. CCU will always code for proline, never anything else, hence unambiguous. It is redundant because CCC, CCU, CCA, CCG all code for the same amino acid, proline. You'll notice that if I change that 4th base, it doesn't really matter so much. I still make proline. So that is the redundancy called wobble. Overall, there are only 4 codons that you should be able to recognize on sight. You will be given the chart in most cases, but it will save you some time if you know that AUG is methionine, which is your start codon, and that you have 3 stop codons, UAA, UAG, and UGA. Further, you need to be aware that there are many types of RNA. We're really only going to deal with 3, but more than 30 exist that we know of so far. The mRNA is composed of codons that code for amino acid sequences and is how DNA information is carried from the nucleus to the ribosome. tRNA is what matches the codons of the mRNA to an amino acid. They have an anticodon at the bottom which matches, is complementary to the codon of the amino acid, and the codon of the mRNA, which carries in the correct amino acid. rRNA is combined with proteins to produce the ribosomes, which you can see at the bottom. They are non-membranous organelle found in all cells made of 2 units, the large and the small. Within the large unit, there are 3 sites for things to do their jobs. The A site is the tRNA and the amino acid coming in to be added to the chain. The P site is where the polypeptide will be made, and the E site is where the tRNA will leave. So as you go through the process of translation, your tRNA will go through all 3 of these sites, the proteins will stay at the P site, and then once everything is done, the protein will be released from the ribosome as the parts split apart. Translation is the process by which the genetic message carried by the mRNA is used to assemble a protein. The mRNA is going to attach to the small subunit of a ribosome using that GTP cap as an energy source. The first actual coded codon will be the one that is for methionine. So the mRNA codon will be AUG, and the tRNA anticodon will be UAC. So that matches. Once that matches, you use the energy to bring in the large subunit of the ribosome, and it's going to form so that the tRNA with methionine is in the peptide spot, the second spot. the P site. Once that is done, the methionine will attach to the ribosome at the P site, and the tRNA will leave. And as it moves to leave, the ribosome will pull in another tRNA into the A site that matches the codon. And that's going to happen again and again and again. So as a new peptide bond forms, the tRNA will be released, leave through the E site, and then new tRNA coming in across. And that process is going to keep going until you reach a termination sequence. When the termination sequence or stop codon is reached, a release factor will bind to the A site. That is going to cause the water molecule to attach to a ribosome, disassociating the bond through hydrolysis. The polypeptide will be released, the ribosome will disassemble, and the protein will begin its folding process. There are some major differences between prokaryotic and eukaryotic transcription and translation, but the big two are that in eukaryotes, you do RNA processing, and prokaryotes you do not. In eukaryotes, transcription happens in the nucleus, translation happens in the cytoplasm, whereas prokaryotes have no nucleus, so everything happens in the cytoplasm. And there is also a time difference. For us, transcription happens, then RNA processing, then translation. It's one after the other. But in prokaryotes, as an mRNA is being transcribed, translation is happening immediately on the new sections that occur. So it's a much faster process because there is no separation, so there is no need for it to be timed separately.